we're going to be doing three things today. First, we're going to follow up a little bit on a few kind of open threads from last week, and this is why we have Adrian Gropper here. Basically, we're going to talk about health comments and specifically about which kind of tools from crypto commerce we can use to make those happen in ways that they're actually privacy preserving. And Adrian will talk a little bit about Trustee and how they're handling self-sovereign identity for this. I'm going to share some info on last week's meeting here in the chat. So this is just to wrap that up. It basically came about from a discussion that we had in which we were like, well, is this relevant? Could we be using self-sovereign identity in some way? I had then reached out to a few people in the crypto space that I know. They had connected me with Adrian, and Adrian is basically doing exactly that. So I'm really, really happy to have you here. I can't wait. Afterwards, we're going to kind of like switch to Matthew Scholz's presentation, and we're going to be discussing the main topic of today, which is senolytic drugs, and if there's a way in which we can accelerate them now in the context of COVID-19. And this was presented by Reason, who is unfortunately cannot be here today, but he was super, super excited and then introduced me to John, and then over John, I got to know Matthew, and I'm so, so happy to have you here to kind of kick us off in this discussion. It was a discussion that kind of like popped up at I think, yeah, really over 5 of the previous meetings already that Foresight had on the topic. So really happy to talk about that. And then lastly, just to kind of prep all of you guys at the end of this, what we already started to do last week is we're going to do a little round of community report outs. So this is kind of to an invitation for all of you guys to get any projects or any organizational updates ready that you want to share with the group. And at that point, you hopefully have a little bit of an understanding of everyone who's in this group. And the idea is basically that we can do a little bit of kind of like sharing and invitations for collaborations at the end of every meeting if it's helpful to you. So that's a new experiment that we're adding. So to get everyone else a little bit more interacting with each other. Okay, good. I've talked much too much. For now, I would love to hear it from Adrian Gropper on self-sovereign identities and specifically what they can do for health comments. And I'm going to share some info on about you, Adrian, in the chat. Okay, take it away.
Okay, so this is a lot of material to cover in a few minutes. So I will touch on 4 or 5 things, and then we could talk about the ones that are worth going more deeply into. Self-sovereign identity is kind of like blockchains themselves, public blockchains. It is a technology that aims to be controlled by the individuals rather than any administrative entity. But on top of self-sovereign identity, we can now build what I would call self-sovereign technology. In other words, technology that, again, is not under the control of any entity, any institution, kind of like Bitcoin itself or Ethereum again. And one of the best and most interesting uses of a self-sovereign technology is self-sovereign health records. So these, again, just like having a cryptocurrency in your wallet, you are in complete control of your health records. It doesn't mean that you can forge them if things come into them that are signed, or if you have a way of controlling access directly to a lab that gave a lab value, then the authenticity of what you control is there, but it is self-sovereign in the sense that only you control who gets to use those. And so that kind of leads into the topic of the day of data commons. It is a very vague concept. I frankly couldn't possibly define what a data commons means. Everybody has their own concept for their own reasons. Commons sounds like such a good word. So what I'm going to try and do is just talk for another 3 or 4 minutes about what you can do once you control health records in order to do whatever it is that, what does it mean to create a data commons? So if you have health records and if you have total control over them with something like Trustee, which is the code base that we work on, then you are in control of policies for access to the health records. And that is a very good thing because people have policies that are entirely private or self-sovereign to them. you don't have to share your policy for why you're not going to give me a quarter. You either give me a quarter or you don't give me a quarter when I ask you, but you're not put in the position of explaining why you made that decision. So we pay attention to the policies for access to self-sovereign health records because those policies are entirely yours and don't have to be put on the wire or shared with anybody. But then we figure out that, okay, who's going to help us choose the policies? You know, our parents help us choose our policies as we grow up. We get older, we might join a congregation that we choose, and then we take 9 out of 10 of their policies and we use them ourselves and teach them to our kids. So in general, it takes too much work to do a fine-grained choice of policies when you have a very rich environment like one's life or one's data set under control. And once you've chosen those policies, now we need to start going in the direction of the commons, of what defines the commons. So one way to start to think of where do I move in the direction of the commons is to say, okay, let's say I get 100 people and they're all going to apply whatever policies they have, and I may not know what they are, to whatever health records they have, and I may or may not know what else they have in those health records, except those things that the policies are willing to expose. Who do we trust to aggregate across, say, 100 patients? Why did I pick 100? Well, if it's 10, it gets pretty easy to tell, you know, who's who, if you're looking from the outside. If you pick 1,000, now you're asking 1,000 people to trust the same aggregator, the same doctor, let's say, or the same clergy person. So, but if I pick 100, then yeah, you know, you might get some stragglers that try and mess with the system, but in general, you've now anonymized or de-identified your particular policies and your particular data, and now you have an aggregate. And then at the last, and this is the end of my commons conversation, at the end of that chain, you now have thousands and maybe tens of thousands of these aggregates of a couple of hundred patients that trust somebody to aggregate their things. And now you're going to generate the real commons that you might be interested in. And for that, you need experts, because the actual individuals that contributed, or even the trusted aggregators like the doctor themselves, they may not have expertise other than the fact that you trust them and, you know, you think that they're worth trusting because you chose them. So on top of all of this, that we started with self-sovereign identity and we went through these 5 or 6 steps that I mentioned, you have to decide if the commons is going to come out with an outcome. Let's say it's a peer-reviewed study in a journal or a product design that people you think are going to buy. Who's going to organize those experts? And how many of those, how will they get interact with the aggregators that are protecting your privacy? And I'll stop there. That's probably about 6 minutes or so.
All right, thanks. I think you've kind of like at least given us the lay of the land here. Are there any immediate questions? Yes, Keith, you already had one.
Yeah, I don't know if this is going to be too blockchain inside baseball, but you were talking about sort of the experts and, you know, who can choose. Other blockchain systems use sort of systems called, I believe, like distributed proof of stake, where like every node in the system can continually elect who these experts would be based on past performance. And then those experts have the ability to validate new blocks in the blockchain and stuff. Is that a potential model for how this could work?
Well, you are right that those inside baseball, it is an inside baseball question. You're right that those systems are voting systems. But I do not consider them experts in the sense that I'm talking about with the commons, right? So in other words, they're only in the trust part. So in other words, if you have a voting system, you can decide to elevate, you know, Dr. Jones over there to be the aggregator at this intermediate level between you and the eventual learnings, the eventual outcome of the commons. That's a vote. The experts I'm talking about are the ones that are actually either going to be the data scientists or the other kinds of scientists that are going to determine how to come out with a valid, what you might consider a valid or a useful result.
Yeah, I guess my question is almost like, is there a way almost to kind of merge those in a sense where somehow
well, there is.
The goodness of what job they're doing can somehow be exposed.
You're absolutely right.
Yeah.
Yes, you are absolutely right. There is, and that technology does not exist. What we call that technology is reputation. Okay? Because nobody says that the expert can't be a citizen or can't be somebody in another country or, credentialed by, somebody you may not even know, but they've proven, that they can do this. And you only have to read the news every day about Facebook and Twitter and social media in general to realize that reputation on a large scale is an impossible problem today. And the technology, which does look a lot like zero knowledge proofs, in my opinion, and if you're a blockchain enthusiast, you know what I'm talking about, will eventually start to crack that nut. But there's nothing out there right now, Keith.
I have a kind of a basic question. Sorry if it's too mundane, but in a system like this, or as you envision it, say I have like every scrap of medical data I've collected about myself in this blockchain, basically. Is there a practical way?
No, it's not a blockchain. I never said it was in a blockchain.
Okay.
Blockchains are not self-sovereign. Identity built on top of blockchains can be self-sovereign if it's done right. We don't put any personal data whatsoever on a blockchain.
Okay, so that's helpful. So I guess the question I was going to is whether or not it was practical to share granular amounts of data from a system like this, or if it was all or nothing effectively in terms of
No, well, that's what I meant by the policies. That's exactly why I said, once you have the data, and by the way, you don't have to collect it all in one big honeypot. If you lose it, you lose the whole thing. You can leave the data in place and use things like encryption or other modern access control methods to share it directly from that place. That way you don't have to bother the source, the lab, for example, to sign it. Because if you trust the lab to hold that result of that blood test, then, and you don't mind that the lab might know when somebody comes looking for it, which they would, then there's no reason to make a copy. Obviously, if you don't trust the lab, well, maybe you should have taken your business elsewhere. So number one, when we talk about these trustee-based health records, we're not collecting everything. We're only asking you to collect the stuff that you don't trust whoever originated it to keep. So in other words, we always pay attention to the excess copies. You don't want your copies going up to the World Health Organization if you can make them stop with your doctor. And you don't want the copies that live in the lab to even come to you if you can trust the lab to control them on your behalf. So that's half of it. But when I said policies, and I said you have to inherit those policies from a community that you choose and a community that's self-sovereign in its own right, that's what I was talking about, is the ability to fine-grain decide who can ask you for a piece of data, and whether you believe who they are, you believe their credentials, you believe what their intent is the way they state it, you believe their reputation. That is extremely fine-grained, and that's why nobody in real life is in a position to say, I'm going to set these 50 switches in this particular way. So you have to inherit the 50 switches, but then you can flip 3 or 4 of them if you decide, that those are the things you care about.
So in our email exchange, you kind of mentioned a few different, let's say, like technical tools, right? And I think to us, what we did in last week's meeting is we discussed a few different types of health commons and different types of structures that would be serving different purposes. So I would love to know from you kind of what is like the one thing you would love such a structure to do? Like what's the kind of like one type of health commons that one could set up with self-sovereign identity that would be incredibly useful right now? And is there particularly an angle in which COVID-19 could be used to make that more likely to happen?
Well, yes, but it's not a clean answer. Okay, so I can't just say, this is the cat's meow here. The thing that I would look at in these comments is what is being learned by whom for whose benefit. So in other words, when you think about a comments, don't think about it in terms of what's the data in it. Where did I get it? What kind of privacy risk is there for me having it? What happens if somebody steals it? Forget about that. Whenever somebody tells you, give me anonymized data because it's not going to affect your privacy, run away. They don't care about you in general. And very rarely will they screw you directly. They're going to screw the other person in your position, or they're going to screw you, somebody like you. So in other words, the societal harm to making inferences about people's data is not to the individual in the form of discrimination or price fixing for that individual. It's in the way of manipulating what we all have access to and how much we pay for it on average. So if you want to take away one thing from this as a definition of the commons, think of the commons as the value of the inferences that result from creating that commons, who gets the value, and what do they do with it? Because they're just as likely to be using it to screw you as they are to be using it for good in the real world. And worrying about the data obscures that fact, whether you're talking about Facebook or Google or any data broker that's sort of collecting data on them, or the government, of course. You never really know. There was a second part to your question.
If you could kind of like swing your magic wand, like what would you want to create a health commons for?
If I could swing my magic wand, I would work in the direction of what's called federated learning or split learning. In other words, keep the data and only control, bring the analytics, bring the things that are analyzing the data further and further out towards the edge, maybe all the way to your own self-sovereign health record. So instead of just inheriting policies from that church group or from your family or whatever, your club, you're now inheriting code that actually does what the commons is supposed to do. And the data doesn't move at all. And again, the reason this is eminently doable, it's not easy. That's why I sort of talk in terms of practically you want to introduce an aggregator that collects 100 people's before.
Is there an organization that already does that? Like, is there a prototype already? Because I was trying to find
context, some. Yes, it's called COVID Safe Paths. It's run out of MIT. You know about.
Yeah, it's contact tracing, right?
Yeah. But the people there, and it is the largest project in the world. It's run as an open source, volunteer operated effort. And the people who started it at the MIT Media Lab are proponents of this thing called split learning. They were doing this 2-3 years earlier. And there's been other work on AI based on something called federated learning, which is roughly the same thing. And I think I sent you links to split and federated learning. And COVID is certainly accelerating that process. But they have not implemented split learning, even though they're the people and they're MIT and all that, they haven't implemented any of that in. And as a matter of fact, if you look at the news yesterday in the New York Times, I think you'll see that Germany and England, both of them introduced technology to do COVID contact tracing, and they have no idea whether it's working or not, even at that level. So in other words, going from managing the data to actually making inferences that are worthwhile, is this working and is it good, is really, really hard.
All right. Thank you so much. I'm hoping that perhaps you can stay on a little bit longer so people can contact you in the chat individually. I also shared some info on the pieces that you wrote here in the chat. And if people want to mull this over more, I'm super happy to talk. There were 2 or 3 more folks in the crypto space interested in actually trying to set up a prototype of such a structure. So if you're interested in that, just reach out to me, and then I can kind of like weave in the conversations. Great, and we have an email address for Adrian as well. Okay, awesome. So now, do you want to say a few last words, or otherwise we'll switch gear now? Okay, good. Okay. I hope you still stay on with us for a little bit at least. And now I'm asking you to rewind and switch gears. Now we're going to come to the topic of today, the main topic, which was suggested by Reason a while ago. He actually suggested it in an official salon that we had, and I think Jim was on that call too. I'm going to share a link to that salon here, which was basically what types of aging treatments could be fast-tracked in the context of COVID-19, and he was suggesting senolytic drugs. It was basically a hypothesis of his that they could produce really good effects if in the context of COVID-19 already. He did say, okay, we have to model this, and Matthew even said, I don't have data on COVID in any way yet, but at least we can maybe kind of reason by analogy. Yeah, I'm super happy to have Matthew here. He was recommended to me by Reason. Really glad to have John Lewis also on the call, who can also probably like get in there and ask a few questions. So I'm not going to take away of any of your speaking time, but I'm going to share the info of the talk now in the chat. All right, welcome, Matthew.
Matt, you're muted.
Can you guys hear me now?
Yeah. Yes.
My computer was complaining about sharing audio for some reason, so I had to grant it permission, I suppose. Oh yeah, thank you very much for having me. I'm very interested in this topic, and it's mostly, I suppose, as a way of accelerating diseases of aging. But I'll provide a little context for what we're doing very quickly in general, and then I'll transition into like how I think it can apply to COVID and some related technologies actually that I think may be worth looking at. And so let's see. So we've basically developed a really kind of new approach to getting rid of unwanted cells, and done this primarily by taking the targeting of medicines, or in this case of cytotoxic treatment, out of the realm of chemistry and into the realm of information. And so what we've done is effectively make logic gates in DNA, like simple if-then statements to kill cells. So instead of building a poison that kills your target cell, we say, hey, cell, are you damaged? Is this pathway on? And if so, kill yourself. And it's quite simple at the beginning, but you can, of course, build very elaborate ones, and you can do other interesting things like modulating the immune system around them. And what this does is it really enables a level of precision that is very hard to achieve by any other means. And some of you who are familiar with us may have seen this video before, but on the left here, we have just the treatment killing P16 positive cells. And this is time-lapse microscopy. They're going into apoptosis, and you can wipe them all out like this if you follow it for a bit. But what's more, I think, important really is the one on the right here, where in the context of aging in particular, and especially in a state where there's comorbidities, you don't want to have collateral damage. Like, if you think of how we kill cells for cancer, the standard of care is, eating poison effectively. There's lots of damage. For aging, that's not acceptable. And also, the cells you want to kill might not be clustered together. Like, you could have one damaged cell in a sea of good cells. And so this showing it in a mixed population where 5% of the cells are P16 positive, you can wipe them out very effectively without harming any of the other cells. And so this is kind of what I mean by we've taken this targeting out of chemistry and into information. They're dying because this gene is active, not for any other reason. And to deliver this requires, a really new kind of technology as well, in that the goal now is to hit effectively every cell in the body. And to do that, you need multiple treatments with something that is both effective and non-toxic and indiscriminate in and of itself. And so this is a technology that we had licensed initially from Entos, the company John Lewis had started, our CSO. And what's special about it is basically these things are non-toxic and indiscriminate. They get into the cell with this little fusogenic peptide, fuses the membrane, and it deposits the DNA or other nucleic acid into the cytoplasm. And I can talk more about that in the Q&A because it's fascinating where it came from and why it works. But this is the idea: indiscriminate delivery, very targeted payload. And for those who've seen what we've done with this in the past, this is just a mouse longevity study where we started treating 2-year-old mice with this once a month, and we have the reds, PBS control, and the blue line is like kind of what you saw from the Baker lab, and these kinds of things where they killed P16 cells transgenically. But what's unique about the approach is that you can change it to target other things. So this black line, we target both P53 and P16, for example. And the results are very striking. Like, you don't have a single death until half of the controls are gone. And even at the end of the study, like, a few of these mice were sacked for reasons that you wouldn't sack a person for, and they didn't all die at the end of the study. Okay, so with this as the context and the kind of approach and our initial focus on Senolytics, why go after age at all? And I think something that probably is understood by this audience but may not be by everyone is that age is really this master corollary of pretty much any malady you could think of. I mean, you take your odds of having cancer, for example, as a clean living 70-year-old are considerably worse than a chain smoking 20-year-old. And things, not to say that your ill-spent youth won't haunt you later, but things really start to fall apart when you're in your 40s, and or after you're in your 40s, probably a little more precisely. But this kind of brings us to COVID, and you've probably noticed already that COVID's not exactly an equal opportunity killer. I mean, it largely spares the young while just slaughtering the old. And as Alex Zavoronokov would say, it's a Gerald-Lavic disease. I like this term he's coined, that it heavily punishes the old. And what's interesting, though, I suppose, is that this ratio of death, in this case in the United States, just recent data I pulled, isn't a whole lot different than all-cause mortality for those range either. So I mean, it's following the same trend as like every other disease that we look at for age. And so I think it's fair to say that what we're really looking at here with COVID-19 is the newest or the latest age-related disease. And I think framing it this way opens up some doors to like really treating the mechanisms of aging more seriously. So as I think everyone would know by now, there's other risk factors and comorbidities for COVID-19 specifically. But I think consider for a moment that all the ones I've underlined there, these are also from CDC's most recent list, all the other ones are underlined ones are already associated with age. In fact, a bunch of them were on that first chart I showed you with everything falling apart with age. And what's interesting is the ones that have asterisks by there's evidence that senescent cells are either a driver or a contributing role of those diseases. There's already this kind of suspicious, I don't want to go so far as to say mechanistic, but at least like something worth looking at, that some of the things that are causing general breakdown with age are actually, maybe more directly increasing your risk factor for COVID-19 than it might appear on the surface. And then I'll go to talk about something I think is worth separating here.
Could I interrupt for just one sec and ask a question or mention something? Or we could just put a pin in this for future discussion. I think you're absolutely right that there's this senescence connection with COVID. But what you didn't write down here, I don't think, but is really interesting that I just discovered, and I'll send you my little speculation and links about it, is that interferon producing cells, particularly the ones, I forget which kind they're called, one type of T cell that produces interferon alpha 2, I forget, it's not really my field. They go massively down as you get older. And in people who have low numbers of these cells and low interferon, COVID just tears through them and kills them. So another connection of senescence to COVID.
Well, and that's actually, I think, a really good intro into this kind of clarification, this distinction between senescence and immunosenescence. I mean, two things that sound similar and are related, indeed, I'd say to a certain extent, but actually have a difference worth paying attention to. in that when you talk about senescent cells, we're referring to a well-defined, well, relatively well-defined, at least extensively defined population of cells that are basically damaged in such a way that they can't re-enter normal cell cycle. And they have these kind of hallmarks, notably of their secretory phenotype, secreting inflammatory cytokines. These are the target of Senolytics and what we're targeting primarily. Immunosenescence gets into a little bit more about what you were talking about, where you have kind of the deterioration of the immune system over time. And it might not be, in fact, I think probably outright senescence plays a fairly minimal role in that. But this is, kind of largely impacts the adaptive immune system. It's driven by things like the involution of the thymus, the skewing of the memory repertoires towards, chronic infections like, cytomegalovirus and this kind of thing. And it leads to, poor response to vaccinations and infections. And the interferon pathway, of course, is, a key driver of innate intercellular response to viruses and, signaling that goes from the intercellular level out to warn other tissues. And so as this thing becomes kind of more energic, basically, it fails across the board. Though I pointed out because it's not something I think we can address with the Senolytic, even though it sounds similar. And I do think that there are things that can address it, and I'll touch on that a little bit in a moment. But in terms of the Senolytics, like Allison said at the beginning and said earlier, there isn't direct data right now of showing the use of a Senolytic for COVID-19. But there is interesting data that's come out from a bunch of these clinical trials, a couple of which have reports already formed about what happens to things like the systemic inflammatory environment after the administration of Senolytics. In this case, mostly in humans, at least, dasatinib and quercetin, the D and Q treatment, as they're called. And I'll point out, one in particular here is IL-6. And IL-6 is a notorious character in the world of cytokine storms, and can be a factor of outright mortality when it spikes. And not only do the Senolytic treatments remove this, but they also specifically seem to target senescent adipose tissue, which is a very kind of, let's say, vocal tissue in the world of molecular signaling. And so the ability to clear the cells that cause the inflammatory environment in general, I think is helpful, both in terms of, they push you closer to dangerous levels on a good day, but also when you consider that these things provide a level of background noise that I think interferes with the immune response in general and overlaps a little bit in this world of immunosenescence. And I'll tie that back in a moment, but before I do, I'll point out that it has been postulated that senescent cells may, in and of themselves, be a preferential target for SARS-CoV-2. And mostly I've seen this suggested due to increased expression of CD26, which certainly was a target for MERS. I think it's debatable if it's used for cell entry with CoV-2, and ACE2, which I'm going to say, I don't think a compelling case has been made for this, but it's probably worth discussing or looking at further. I just think that the authors of the paper I cite here on it, I don't think the papers that they cite make as supporting of cases they might have liked. But I guess to tie it together, and why I think one of the better pieces of evidence that a Senolytic may indeed be helpful in this is actually from kind of a class of drugs we might call Senomorphics, and notably rapamycin and its analogs or rapalogs. And so these have been shown in humans to improve response to both vaccinations and infections. And so, but the premise they work on, that these target mTOR, is a well-known pathway for immunosuppression. And so it is slightly, paradoxical that suppressing the immune system might lead to better response to a pathogen. Indeed, it is very much, I think, a hermetic phenomena. If the dose goes too high, it will not be helpful anymore. But at the lower doses, it does seem to improve it. And I think one of the mechanisms by which it improves it is effectively lowering the background noise. A lot of thought in the world of senescence has gone into, why do NK cells, for example, stop clearing senescent cells as you age? And think one of the reasons is that as the environment becomes more and more inflammatory, it becomes difficult for your body to distinguish the signal from the noise. And so tools that are able to reduce that baseline inflammatory environment lead to more precise adaptive targeting. And I think this is what, or at least a part of in things like rapamycin, and also some of the benefit that a senolytic might have with it. I guess I'll stop there because I don't know if that's 5 minutes or not, but it seems like enough. So, throw it back into discussion land.
Great. Any questions?
Yeah, I have a quick question. It's Robert Zim speaking. I'm not an expert on viruses, but wouldn't all coronaviruses and flus affect older people more so than younger people? And so this is kind of a misleading argument for that slide that you showed.
I mean, yes, I would say though that it's not at all misleading, and that in general. So flu is interesting in that often you'll see like kind of U-shaped mortality where like very young get killed by things and very old, but the middle are safe, and that's not strictly true. But I think as a general rule, pathogens take a bigger toll on old people than young people. And so the reason I say I don't think this is misleading is that there's got to be a reason for that, right? It's not just a coincidence. So what we're kind of looking at here is how can you untangle what the driver is behind that? Like, and is it possible that some of the accumulation of damage throughout the body, in particular the increased inflammatory environment in the body as you age, might actually be causal, or at least a contributor to the diminished response with age to viruses? Does that make sense, or did I address that?
Yeah, I guess, yeah. I'm just saying, like, it seems to me rather, speaking from the point of view of sort of a person from the general mainstream, it seems rather opportunistic to say that, COVID is the reason we should fund aging, as opposed to just about any other disease that affects older people more.
It's certainly opportunistic. I mean, I think that's actually kind of a stated purpose in this respect. There are better things you could target than COVID, but when the money's flowing, grab a bucket. So this is not, COVID is not the indication I would choose for aging. In fact, it's not the indication we've chosen. We're going after chronic kidney disease as our first target for a senolytic normally. The topic of this conversation,
right,
was, could a senolytic play a role in it? And I think it's possible, although the data is far from certain on it. Although if we could use the opportunity to advance it, I'm all for it.
Thank you. I think Sebastian.
So playing devil's advocate, you think that some people?
Yeah, I'll just jump in. Really interesting stuff from Oisin as usual. Thanks, guys, for that. I'm a bit of a stickler on the precise use of language sometimes, and with rapamycin, its various geroprotective effects are often conflated. So there's the anti-inflammatory effect, the translation inhibition effect, and the autophagy enhancement effect, and maybe some others we're not aware of because mTOR is such a central node of metabolism. But I guess I can speak with a little bit of authority because I and some colleagues created this company, Aovian Pharma, a few years ago with Dudley Lamming to actually divorce some of these effects of rapamycin by targeting mTOR complex 1 instead of both complex 1 and complex 2. And many of the deleterious effects that you'd like to avoid in a geroprotector actually emanate from hitting complex 2. So the point is, the immune response enhancing effects of rapamycin may actually be unrelated to the immune suppressive effects that we see and that it's approved for. In fact, I would argue that its autophagy enhancement, the autophagy enhancement effect is critical. And this is also known to be critical for the life extension effect. So this is why we sought to, and apparently have succeeded, in creating an analog of rapamycin that retains the autophagy enhancing effects while divorcing them from the immune suppressing effects. So time will tell whether it still enhances the vaccine response.
I think that's totally fair. And in fact, to be clear, the low dose rapamycin treatments are, effectively thought to, at lower doses, hit mTOR1 harder than mTOR2, like so that it takes a higher concentration to hit mTOR2, and used for, organ transplant, it's a high enough dose to hit both. But in the studies that I was citing, it was either, in some cases, they are rapalogs engineered to target mTOR1 also, but done at a low enough dose that hitting mTOR2 in theory was minimized. But I think you're absolutely right in that the complexity of that pathway is staggering, really. And I wouldn't pretend to be an expert on it. The thing that had caught my eye, and the reason I mentioned it in this context, was actually the more the anti-inflammatory aspects of it that seem to be prevalent even at the lower doses as well. That you don't get it hammered as badly on immune suppression, but you do see reduction in inflammation. And that the general thought, and I haven't found good data on this really, or like at least exhaustive, and if someone has it, it'd be interesting, is to really get a good comparison of the cytokine profile changes from SASP versus lower dose rapamycin or mTORC1 inhibition specifically. So I think that they may not be as similar as it appears superficially, but certainly there's considerable inconsistency in the data coming out from Senolytics, at least in the sense of some don't have it at all. And so, but IL-6 is and IL-1 are both pretty common, I'd say.
Yeah, for sure. IL-6, IL-8, IL-1. Yeah, good points. I don't want to get too in the weeds, but using an immune suppressant like rapamycin or one of the mTOR inhibitors or RTB101, which is the Restore biomolecule, I think that's irresponsible, because you're talking about giving this to a patient population that already has compromised immune function. And you're hoping that you're going to either enhance the immune response or you're going to inhibit the SREBP2 lipid formation process and viral replication, as was kind of the hypothesis for Restore Bio, which has failed.
I mean, it is worth considering that the use of corticosteroids, which are a far bigger hammer on the immune system, at least for short-term high dose, are not quite standard of care, but quite commonly used to deal with the inflammation aspect, specifically in the lungs and in a ton of other infections for that matter. So I wouldn't go so far as to say it's irresponsible, although I definitely would exercise caution with this. And I don't think that it would be prudent to simply throw a bunch of people on low-dose rapamycin and see what happens necessarily, especially if they're already dysfunctional. But yeah, I mean, there's definitely a track record for using immunosuppressants in the context of an active infection.
Yeah, when it's an active infection, you're going to die if you don't suppress the cytokine storm, for sure. But if we're talking about giving a rapalog to patients who are not yet sick to enhance their vaccine response, when there are plenty of other adjuvants that do that, or you want to give it to patients prophylactically, as Restore did in respiratory tract infections. That's a prophylaxis model, right? So you're suppressing the immune system and then hoping that they get less infections. It makes perfect sense to use corticosteroids once the patient is, in the ICU and on death's door. Of course, there is a trade-off because you are suppressing the antiviral response at that point. But anyway, but you do bring up a really good point, which is rapamycin and rapalogs, they prevent the occurrence of senescence. And when you have a lot of senescent cells, they also suppress the SASP. That could be the translation inhibition mechanism. We're actually looking at a molecule called minocycline that works quite similarly. That's sort of one of the newer geroprotectors out there. And so what you would want to do is give a non-immunosuppressive autophagy enhancer / translation inhibitor / anti-inflammatory to lessen the incidence of senescence, to reduce the senescent load. But once once the senescent cells are already there, of course, you need an approach like what your company has to get rid of them. And I personally work with rapamycin in the lab, and it doesn't kill senescent cells, it just suppresses their growth, at least in vitro. So anyway, that's why your company is so important.
Yeah, it's all hands going up. I don't want to go.
I think
You want to go wrong?
Keith wanted to bring up a new point. So I think if Creon, if yours is towards this particular point, then you go first. Gotta.
I think it is. I have a small write-up that I want to send out, but it's too big for the text chat, so maybe I'll just email it to the presenter. But there's a deep set of connections, right, between inflammation, interferons, and the progress of viruses in the body, particularly COVID, which seems to actually have adapted to mess up the interferon pathways in infected cells. But even without that part of the arms race, immunosenescence, I know you tried to separate immunosenescence from senescence, but inflammation is a driver of senescence, if I'm not mistaken, and a driver of immunosenescence, and that drives down interferon production by screwing up the dendritic cells, the plasmacytoid dendritic cells, and then that just makes it ineffective to mount a defense against viruses, because that's the bridge cells between the innate immune system and the adaptive immune system. And so immunosenescence driven by inflammation messes that process up, and now both the innate immune system and the adaptive immune system get dysfunctional in inflamed people and old people who are, of course, overlapping cohorts.
Yeah, in the interest of time, I don't think I can address all that, but I would that short-circuiting the interferon response is an oldie but a goodie in terms of the bag of tricks viruses like to play. Lots of them do it, and it's very helpful for the first steps of viral replication. As they once they're in a cell, beginning their hostile takeover. But the reason I wasn't going too far into immunosenescence wasn't because I don't think it's relevant. It's just that a Senolytic can't directly address it. At least I don't think it can. And not that it wouldn't have some impact on it, but it was just I was more off topic for my 5 minutes. That's all. I wasn't trying to ignore it entirely.
Okay, sweet. Keith, you go for it, and then we have another comment, and then I would love to drive back home. Maybe you can already start thinking about this, Matthew, but what kind of changed in relationship with COVID-19 and your respective projects, and how can we leverage it actually to kind of advance analytics and or to advance several, at least, hypotheses that we have at this point?
Sure. Well, hopefully my comment will tee that up a little bit. I wanted to respond a little bit to Robert's earlier devil's advocacy about it being opportunistic to talk about COVID as a disease of aging. And I think that's a fair comment, and we definitely have to be tactful in how we do that. But I think it is extremely valuable to play up that angle, and is a legitimate one. And at least from my personal experience, I found it to be very effective, and I'm not really getting any pushback when I bring that up. And I think we can almost use this crisis, I don't mean it in that way, but to make a transition that I think we need to make to really drive this field forward. We typically bring some statistics up that you guys have probably all heard a million times, like over 100,000 people, die due to diseases of aging. And then it's kind of like Alzheimer's, heart disease, cancer, and then there's all the other diseases and getting hit by car accidents and stuff, right? But I think that there's really an angle to say, here is this number that's based on those three things. But what does that number become when we add COVID? What does that number become when we add the flu? What does that number become when we add all these things? Because they are certainly age-related diseases. And I think there's a way that we can really be in this current era, transition to that and make it clear that almost everything really is an age-related disease, unless getting hit by a car, unless, the decreased driving ability.
Testosterone-related disease. bad driving when you're a teenager.
Yeah, I actually think
it's an age-related disease. Sorry, it's an age-related disease, but it's infectious, which is a very big distinction between the age-related most people think about.
Well, I'll actually agree with you and say that that particularly is why it's so useful as a tactic now, because just psychologically, right? If you talk about cancer, people are very easily able to employ cognitive biases like, hyperbolic discounting and stuff like, oh, that's like a disease when I'm 90. I don't need to think about that. It's invisible. But when you tie the biology of aging to something that is attacking you, right? That makes people want to do something about it, and it makes them much more impelled to action. So I agree with you, and in a sense, that's why it's a very powerful tactic. That makes sense? Yeah. So
I think we had Ronald who had his hand up earlier to make a direct comment to this before we take another one. Ronald, are you here?
Ronald?
Yes. Hello.
Just very quickly, I sent a comment around on the difference between cellular senescence and immune senescence. You think of the immune system as an organ. It's loss of function of an organ. Cellular senescence is the change in cell fate that affects the tissues and organs. So they're, as pointed out, they're different. Just the framework.
Right. No, I do think that the language here is important, and it's good to get it right. Thank you so much. It was like a good pop-up, when you like hover over something, you double click on it. So thank you so much for that. Okay, next one up we had, I think, Vargas with a comment over here. So if you'd like to unmute yourself, go for it now.
Hi, yeah, I'm just wondering about the age-old question with Senolytics, I guess, which is about targeting the right cell types, because it doesn't really seem clear to me that all senescent cells actually are bad, or that we necessarily want to kill all senescent cells. I think Judith Campisi published a paper recently with lung cells that seemed protective and were senescent cells, and the paper I linked by Dimitri also mentions cells that seem quite important that are senescent cells in the liver that are bad for the mouse when they're cleared.
Well, I think the Dimitri one is very questionable, but partly due to the way they're killing senescent cells and the drug they're using to activate that and its anti-angiogenic properties. So I'd say actually to back up that one well-known use of senescent cells is in wound healing. And so ablating the senescent pathway per se would almost certainly be deleterious. Like I don't think anyone in the field suggesting it. We're looking at like point in time, hit and run type approaches where you go in there and you clear some out and then you go away and let it return to normal. And so I think one of the reasons Dimitri saw the data is because he kept poisoning the liver while he was hurting the liver. So he's destroying senescent cells and causing injury concurrently. And I think it's not a coincidence that he's the only person on the planet who's ever seen that. And like literally every other study that looks at senescent cells doesn't show that. But whether they be things like ours that slam the liver like a truck, or any other tool really that can pick those things off. The liver is an easy target to hit with a senolytic. And so but to the extent that, there could be, senescent cells at any given moment in time that you don't really want to kill is, I suppose it would be impossible to have no collateral damage, but like most of the data, we've seen shows that these provide meaningful benefits. So if there was a harm that was caused, it's dwarfed considerably by the benefit. I suppose the final kind of more philosophical question is, have we really defined all the senescent cell populations in the body? And I'd say no, actually. Like that, and I think most people, if you look at like papers coming out from like Marco DeMaria and these guys, where they're trying to figure out how do you even definitively pin down what a senescent cell is, it quickly becomes complicated. And I suspect we have not yet characterized every senescent population in the body. But that's not to say that there isn't meaningful benefit that can be achieved through some of these tools to ablate, say, the P16 cells or targeting beta-gal or those kinds of things. And I think what we're looking at here is kind of like the relatively early generations of senolytic treatments. And ours is quite unique in that it's on a genetic pathway specifically. It's a P16 and P53. But what we saw that wasn't immediately intuitive is that there's considerable synergistic effect between the P53 and the P16 killing. And you could argue that the D and Q treatment kind of suggests this as well. But in that case, it's, the Q is far more lousy than the D, and the D hits P53 far harder than anything else. And so there's an argument that both are needed to function as a senolytic due to kind of, Kirkland would say, redundancies in these pathways. But I think that there's a lot that could be gained from the tools that we have today. And I think that it'd be a disservice to not push them forward. But it's not to say that there won't be better ones later.
So what do you think of kind of next steps from this? Do you think that there's anything in particular that you'd like to investigate next? And perhaps.
Well,
right.
Well, I guess we're in kind of an odd spot in that we're using the same delivery technology to build an outright COVID vaccine, and so we're in our COVID stuff. We're playing much more directly at COVID. But one thing I think is particularly promising that kind of circumvents these problems is the ability to use tools like this for vectored immunoprophylaxis. So although our vaccines for COVID are DNA based, we just express viral proteins in the cells and the body targets them. And it's nice because you get the T cell targeting, I think, a lot better than you would from just injecting protein subunits with an adjuvant. But and it to a certain extent minimizes the need to run through some of these steps and the various toxicities you could get with the higher doses of adjuvants in people. But the vectored immunoprophylaxis kind of short circuits this entirely. And we did this as kind of a biohacker project with monkeys years ago, where we got the ZMAP antibodies for Ebola during the Ebola outbreak. And we modified them for mammalian expression and electroporated them into the muscle of monkeys. And for a couple $100 for the DNA, we got a few $100,000 worth of ZMAP running through the veins of the animal. And the beauty of that is that you effectively taken a good chunk of the immune system out of the equation. Like you're basically just, relying on high doses or high titers of antibodies to minimize the number of virons that can start a productive infection. And I think those kinds of tools might be more helpful in the populations that need them the most. Because the one concern I have is every day it seems we're getting new data from new vaccine candidates that are out in the press. And the data runs from, not very compelling to reasonable, I think. But the fact is they're all testing these on either healthy patients, young patients, or monkeys. And even with the seasonal flu vaccine, the dose required to elicit a response in a geriatric population is way higher. And so, it's probably not a coincidence that we've never had an approved coronavirus vaccine, despite things like MERS and SARS and concerted efforts to get them. And so these nucleic acid technologies, I think, definitely give us a new tool to swing at. But the more we can either improve the immune system or circumvent it to a certain extent, I think the better in the short term.
All right. If I channel Reason for a moment, he basically said, what we need a model for is the following. The basic point is that lower inflammaging might mean lesser ulcerative cytokine storm, and the comorbidities raising risk may be doing so because they raise chronic inflammation, but this is by no means demonstrated to be true or viable. It needs testing in a model. So could you come up with a model or a way in which that could actually be tested? Now I'll post this here.
Yeah, the easiest way, I suppose, would be to take a bunch of either old monkeys or old ferrets, give them a senolytic, and then challenge them with the virus, or maybe the senolytic and a vaccine and then the virus. That's, I guess, the basic academic way to do it. It'd be pretty straightforward. The time it would take would be considerable, though, in the context of this particular outbreak. But one thing that I think could be explored potentially is you now have a fair number of patients who have been treated with senolytics, in the context of clinical trials that are documented. I would be very curious to follow those patients through the COVID outbreak and see how they fare if they get sick.
Ocean's too busy doing their work because they're a small startup, but who's talking to Kirkland about this, right? All the Mayo trials.
Yeah, I mean, this is how we got the data initially from the rapamycin stuff, is they're noticing this in people who are taking it, and they did dedicated trials later to try to tease it out, but with the seasonal flu vaccine. But I mean, the other way one could do it, if you're into the more community biohacking thing, is we have easily accessible Senolytics, and we also have easily accessible vaccines for like flu or tetanus or things like that. And so you could basically run a whole bunch of people who basically, some of them take the Senolytic and then take a vaccine, and some of them don't and then take a vaccine, and measure the responses. And it's cheap and easy to do. I don't think it would earn you the respect of your academic peers so much, but it would actually be very interesting to see it. And it's quite feasible. The cost is low enough that, I mean, D and Q, especially if you get the D from India, it doesn't cost very much. And a tetanus vaccine or a flu vaccine doesn't cost very much. And so if you have a way of aggregating the data, you could actually run a pilot trial with probably on your group. And you just want to age match enough of them and get before and after on cytokine profiling and all that good stuff.
Matt, why a vaccine? The vaccine's not going to induce a cytokine storm.
Oh, no, but the response to the vaccine seems to by the existing levels of these. So I mean, to intentionally give people a cytokine storm is a little dicey for ethical reasons. But,
no, but there's enough people who've probably been exposed to actual COVID infection who were in clinical trials for Senolytics that you might be able to look at differences.
And that'd be my first choice, because they've already had all the venom. So the question is, you'd have to go to the patients to get their consent again for this, which would require like going through because presumably Kirkland doesn't know who these people are. Like they had to be done through a third party, but someone can reach out to them. And given the COVID pandemic, they'd probably be receptive to it and say, hey, we'd like to follow you because we gave you this treatment. And I mean, it wouldn't be a huge data set, but they'd all be older, which is nice. I mean, I think everyone on those studies they're all age type diseases, so they should be old anyway. And I think that's like the lowest hanging fruit to see if a Senolytic might work, is to simply look at the data that exists and follow these people out for a bit and see what happens.
Well, okay, so there's like, I guess, two strategies, right? One would be, again, like health comments, let's do it ourselves. And the other one would be, okay, let's try to actually reach out to someone. Well, if someone points me in the right direction, I'm happy to do that. Or I'm happy to help facilitate it if it's useful at all. Are there any other questions to Matthew? Any comments? Okay, good. Well, thank you so much. This was totally fantastic. I got a lot of really kind of like very sad faces for people that had to leave early. Thank you so much. I really appreciate it. I'm hoping that you will send me your slides. That would be fantastic. And yeah, okay, great. Super.
Yeah, no
quite informative. Okay, very nice. Okay, for the last few minutes, for those who are still interested in doing this, I would love to know, is there anything new that is popping up in your research, is popping up in your company that would help from exposure in this group, either as a pair of eyes or like many pairs of eyes on it? Is there any invitation to collaboration, any updates that you want to share with this group? Feel free to raise your hand, and you have basically 30 seconds to just say, okay, John Ferber, you're first, then Jim.
My question actually is for Matt. When will the Ocean products be available for human testing?
Oh, well, I suppose that can go into my update. We had our first regulatory hearing for the oncology product just like a month ago. And so the goal is to have that thing in clinic within a year, funding dependent, of course. So that targets P53. The P16 one, we're going after chronic kidney disease, and the goal is to have pre-IND hearing for that in the spring of next year. And we'll have done GLP talks already, so we could enter it, and conceivably as early as summer or fall of next year. And the goal for us on the strictly aging side is you get both these things into the market, or into at least human trials for specific indications down the kind of center of the fairway for FDA. and then they, of course, can be applied to other things, both broadening the label and combining them as it goes on. Because, I mean, the data suggests that taking both together is optimal. And what we'll do eventually is we'll build a combined construct that's a single new drug, basically, with both of them built into one. But that one will take longer. And I know a lot of people are very eager to get their hands on it in a relatively early state. And so I think the first bar is get them in clinic. I mean, once there's an open IND for both, you have a whole lot more tools at your disposal to apply them in different areas.
Jim's next.
Yeah, so this should be of interest to people. Carl Flegger, who's actually on the call and might want to say something, has a wonderful summary paper about vitamin D. So vitamin D deficiency is very widespread. It's linked to severity of COVID. It's also linked to depression. It's also linked to lots of pathologies of aging, like cancer, cardiovascular disease, diabetes. I would encourage everyone to read his paper, which I'll post in the comments, and Carl himself might want to say something. Or he might have just left.
I don't know how much that's to be considered in scope for these Foresight talks, but I think the data is compelling, and it's in some ways a lot like the data for masks, when masks were not being highly recommended widely, and a lot of people thought that societies and governments were missing a big weapon to help tame the pandemic by not advocating for masks more severely. And clearly the inflection point for masks came a couple months ago. And though there are obviously holdouts who don't believe it, there's the sort of general consensus now is pro-masks. And I think the evidence for vitamin D is actually even stronger. And I've tried to write it all out in a very user-friendly way in a full 25-ish page review, but then also there's a one-page summary for the people who want the quick summary, and also a list of things that I think decision makers and public health people and hospitals and FDA should do. And check out the one-pager, the one that has the facts and the link. It's pretty simple.
All right, Vargas, you're next.
Hi, just really quickly, we've recently published the CellAge database of genes that can induce or inhibit senescence in genome biology. So I've just linked it. And if anyone has any questions or comments, we can talk about it. Basically, we just look at genes that... Sorry?
Please say a few words about it.
Yeah, basically... the database is built based on in vitro genetic manipulations in human cell lines, and we classify genes as whether they can induce or inhibit senescence, and we look at replicative and stress-induced and oncogene-induced senescence, not just one type of senescence. We look at all three. But yeah, so the database is up now on João Pedro's Hagar website, which I'll link as well. So hopefully that'll be useful for any Senolytics research as well.
Thank you so much. Okay, Christine, you're next.
Hi, three of us in this group, I'm not sure if the other two are on the call. They are Brian Delaney and Hong Zheng, I think, are starting a spreadsheet where we compare exosome providers based on quality and safety. This is being done both to recommend to various groups for our own personal use if we want to use exosomes, and also it's not primarily designed for use by investors, but it would be useful if it were done well for investors, I think. So anyone who wants to contribute information to that, that would be appreciated.
I can send the link. I can send it in the chat if I know how to do that. If not, I'll send it by email.
Sounds great. Marcus next, then Keith.
Hey, hi everyone. I'm hesitant to share this because I'm just, as a scientist, but this group I think can take things with a grain of salt. I've been going down a rabbit hole of flavonoids in COVID, and bicholine is interesting. So we have some I think there's growing evidence for perhaps its anti-ferrin activities, but also some maybe biohacking evidence. For choline, you can find it in Chinese skull camp. So if you're just doing research into the there's also some evidence in H1N1 for it. There was recently something too, also bicholine plus quercetin, but I think like just there's something worth it there. And Christine, so we're just licensing a autologous exosome tech to like the PRP to a PRP industry. So if you are going down exosomes, they're very potent molecules. Potent not molecules, but a package of molecules. So I'm very skeptical of allogeneic exosomes. But so anyway, feel free to hit me up. We have an autologous tech that we're using for autologous exosomes. I'm not sure what you would want to be using them for, but yeah.
Great.
Very cool. That may even want a future meeting. Now, so I'm going to get on it too. Okay.
Sure. Yeah, I just wanted to give a quick reminder that our annual conference is coming up on August 20th and 21st, online this year for obvious reasons. Information is in the chat. It would be great to see everyone there. And also I have a sort of request for help. One conversation that I'm kind of having with people like the NIH and stuff, something I'm trying to figure out is, are there any grant opportunities, not just for the biology of aging itself, but also for public outreach involving that? Because some of our initiatives are starting to hit millions of people, videos and stuff that we're doing. And if we had grant resources to siphon into that, we could probably switch that up to the tens and hundreds of millions of people. But I find it I very rarely see any organizations that are funding like the outreach advocacy side. So if anyone happens to know any organizations that are doing that sort of grants, definitely let me know.
Lovely. Am I missing anyone? I want to thank you so much, Matthew, for joining today. This is really, really fantastic. And I'm hoping to see many of you next Friday. I'm going to send out an email announcing next Friday's meeting. Carl, you got your way, and I'm happy to announce that I was successful in valuing the tubes that you recommended. And next week, what we're going to be discussing is aging clocks in the context of COVID-19. And we're going to have Vadim Gladishek, who will be speaking on COVID-19 as a disease of aging and an opportunity to intervene. And we're going to have Steve Horwath speaking on epigenetic clocks for clinical and preclinical studies. And we are going to launch into a discussion all focused on how we can advance different types of aging clocks in the context of COVID-19. And I think this has been suggested by a few of you now, and I reached out to Carl, and Carl made those recommendations. So thank you so much for that. And I can't wait to see you next week for that. And I also scheduled the one after, but I'll tell you next week about it. But for now, thank you so much. This was fantastic. I hope you have a lovely Friday, and I see you next Friday.